Hello, everyone. Today we're going to talk about muscle tissue and organization. So we're starting the muscular system. Now I have a um, pleasure introducing you to this system because I've actually studied this system for a while during my PhD. So towards the end, I'm actually going to give you an idea of what I was doing during my PhD, or at least some of it. First of all, what we need to look at is the muscle tissue. So muscular system comprehends um, over 700 skeletal muscles. But when we talk about muscle, we cannot talk only about skeletal muscle. You're going to see that there are actually three different types of muscles, and we find these in different places. If we focus only on the skeletal muscle, then all the skeletal muscles together form the muscular system. Skeletal muscles are organs. They are made by different types of tissue. We have epithelial tissue, we have connective tissue, muscular tissue, and nervous tissue. The epithelial tissue, we find it in the blood vessels. Blood vessels that are coming into the muscle are lined with endothelial cells, which are a type of epithelial tissue. There's a lot of connective tissue that it's keeping the muscle together, and we're going to see that in the structure of the muscles. Of course, they're made mainly by muscular tissue. And then there's also nervous tissue that helps the muscle do contraction, so movement. These skeletal muscles are cells are called muscle fibers because they're actually a fusion of different cells. When these cells are stimulated, then the internal changes that happen will make the muscle become shorter, and we call these a contraction. When the muscle relax, then it's stretching out. The result of these shortening is movement. Not only the shortening, but also the stretching. So the relaxation will also lead to the opposite movement compared to the shortening. So as you can see here, this muscle is contracting. And so what you're doing is flexion of the forearm. And then when the muscle is relaxing and the opposite muscle is contracting, then you have extension of the forearm. Now, muscle tissue can be classified into skeletal muscle. These are all the muscles that allow us to move. But we also have cardiac muscle tissue that we talked about when we did the cardiovascular system. This is the muscle that pumps the blood out of the heart. And we also have smooth muscle tissue, and you might have seen these over and over in different organs when we talk about layers of smooth muscle tissue that help contracting or relaxing certain blood vessels, for example, or airways or intestines. Those were a type of smooth muscle tissue that is involuntary. So we're going to start with skeletal muscle tissue. Here you have an histological picture of it. This type of tissue, muscle tissue, is attached to the bones of the skeleton. So the skeletal muscles that are made by skeletal muscle tissue are attached to the skeleton, and by being attached to the bones, they allow the movement. This is a type of voluntary muscle tissue. What that means is that we can control it through our nervous system. We can voluntarily control it and make it contract or relax whenever we want. The skeletal muscle fibers that make this muscle tissue are very long, parallel fibers. You can see that they have multiple nuclei. These are 1, 2, 3, 4, 5, 6, 7, 8, and more nuclei in just one muscle fiber, which is considered one cell. So these cells are multinucleated. They have multiple nuclei. They also have striation, so they are called striated. You can see this striation. The striation are perpendicular to the muscle fiber. So these small lines that you see here are the striations of the skeletal muscle tissue. So these are the three main characteristics, and that's how you can identify skeletal muscle tissue. Parallel muscle fibers that are very long, that have multiple nuclei, and that have striations. When we look at cardiac muscle tissue, it's a little bit different because these are not long fibers. They are also not very parallel to each other because they actually branch out. They also don't have multiple nuclei. So there's only one nucleus per cardiac muscle cell. these cardiac muscle cells are also called cardiomyocytes. So these cells are Y-shaped. So if you look at this cell here, it goes one way, and then you see there's another little region next to it. You can see these in other ones. You can see that in the diagram. So Y-shaped cells with only one nucleus. They still have those striations. If you look closely at this picture, you will see these perpendicular lines that are these striations. There are also these very thick lines in between cells, and these are the intercalated discs. If you remember from the cardiovascular system, these intercalated discs are the region where there is gap junctions, and at the same time also cellular junctions that are keeping the cells together. So there's a mechanical and an electrical communication between the cells. So these are the main features of the cardiac muscle tissue when you look at it from a histological point of view. Y-shaped cells, only one nucleus. They have a striation, and then they have these thick lines here called intercalated discs, which you don't find in the skeletal muscle. Now, this type of tissue is involuntary, so we cannot control the beats of the heart, and it's autorhythmic. And what that means is that there are pacemakers within the cardiac muscle tissue that allows these cells to contract and relax. And remember, there is also the autonomic nervous system that controls the activity of the heart by increasing or slowing down the heart rate. But that's also involuntary. And the last type of muscle tissue is smooth muscle tissue. These are single nucleated cells. So cells are very short. They have one nucleus per cell. When you look at it, you actually don't really see the boundaries between cells. You just see sort of it almost looks like a dense regular connective tissue, but with nuclei all over. Okay, so this is a smooth muscle tissue. So the cells are so close together that you cannot really see the boundaries of them. But the thing that makes it different from the other type of muscle tissue is that it does not have any striations. If you look closely, this is how the cells are parallel to each other. So perpendicular to it, there are no striations. So it's not striated. Cells are very short. There's no intercalated discs, and they are parallel to each other. We find this smooth muscle tissue in a lot of the internal organs. It's usually a layer of muscle, for example, in the muscularis, in the tunica media. So it forms layers of the stomach, of the intestines, of the urinary bladder, of the blood vessels, of the airways, and so on. Now, the contraction of the smooth muscle is involuntary, so we cannot control it. It's controlled by the autonomic nervous system. And these are the three types of muscle tissues side by side so that you can compare the histology, you can compare the features of them. Remember, the skeletal is voluntary, cardiac and smooth are involuntary. Skeletal and cardiac have striations, while the smooth does not. The cardiac and the smooth are a single nuclei, while the skeletal has multiple nuclei. Now, for the purpose of these, well, first of all, let's talk about the property of muscle tissue in general. So all these muscle tissues, they have excitability, which means that they're able to respond to stimuli. They are able to also transmit electrical activity, electrical signals along the cell membrane, and this is called conductivity. So they're able to conduct, to carry the electrical signals. They're also able to generate tension and shorten the cell length, and this we call contractility. So they are able to contract. At the same time, they are able to extend, to stretch out, and this is called extensibility. And then they are also elastic. They're able to return to their resting length after they shorten. Now, this is a little bit different than being stretched out beyond the resting length, because when you stretch out, when they are at the resting length, it means that they went back to the original shape. So that is elasticity. While extensibility means that they can actually be stretched out more than that, and that's extensibility. Now, for the purpose of this unit, we're actually going to focus more on skeletal muscle. So these are the functions that are specific to skeletal muscle. First of all, body movement. This is a type of voluntary movement. We decide when to contract or relax these muscles, and that allow us to move, to produce movement, to walk, run, crawl, to jump up and down, and so on. But it also maintains posture. So even if you're not moving, you're maintaining posture of your entire body. So for example, I'm sitting here, I'm still using my muscles to keep my back straight, for example, my head tall, and so on. And so they maintain posture. They also help with protection and support of other organs. They regulate elimination of the materials. This is mainly for, I should have taken that out actually. This is mainly for a smooth muscle, but there are some sphincters, for example, the external sphincters that we've seen in the urethra, also in the anal canal. Those external sphincters are actually skeletal muscle. So for that reason, we have this function here. They also are able to produce heat by contracting and relaxing. These these muscle fibers are producing energy, and they are also producing heat from that energy. And so they help maintain the body temperature. You can see these here in this picture, which uses a camera that can detect heat. This is a person running a bike, and you can see that they are warmed up by the use of these muscles. So that's why they look red. Okay, so one other way to generate heat is actually when muscles are contracting, when you're shaking because you're cold. That's situation that happens because the muscles are trying to generate heat by contracting and relaxing. So the shaking that you have when you're cold is actually the muscles trying to compensate for being cold and try to generate some heat. In terms of the structure organization of skeletal muscle, we have a muscle here, which we're going to see that it's actually covered by other things, and specifically connective tissue. But here we're going to focus on the muscle tissue. So skeletal muscles are divided into these bundles, which are called fascicles. So if you take just one muscle and you cut it across, you're going to see that there are these bundles of muscle fibers. And the bundle itself is called a fascicle. So skeletal muscle are made by multiple fascicles. And each fascicle is made by multiple muscle fibers. And remember that the muscle fiber is the cell of this tissue. Within that cell, we have filaments, and these are basically proteins that make these myofibrils. The myofibrils are really important because those are the units that are actually contracting in the muscle. And so the contraction of this myofibril leads to the entire muscle contracting. The myofibrils are made by myofilaments, which are basically strands of proteins. So we have muscle fascicles, and then muscle fibers made by myofibrils, which are made by myofilaments. Remember that myo means muscle. Okay, this is another picture showing you the same structure. We have a muscle, we have a fascicle. Within the fascicle, we have a muscle fiber. Within a muscle fiber, we have those myofibrils. Now, what we're looking at is what is dividing all these structures. So we have connective tissue that surrounds all of these structures. So the one that surrounds the muscle itself is called epimysium. Epi means above, myo means the muscle. So this is the connective tissue that is above the muscle, and it's separating one muscle with another one. There are regions in which we have multiple muscles close to each other. And so this epimysium is separating and insulating the muscle so that when that muscle is contracting, no other muscle are contracting unless they are also elicited by nervous stimulation. Then within the muscle, we have fascicles, which are covered by a layer of connective tissue called perimysium. Perimysium, it means around the muscle and contains blood vessels and nerves. So if you open up the perimysium, you're going to see blood vessels and nerves that are leading into the muscle fibers. And then if we take each individual muscle fiber, it's also covered by a layer of connective tissue. And this connective tissue is called endomysium. Endo means inside. So basically, for this level of organization, we have the endomysium covering the cell membrane of a muscle fiber. Okay, so endomysium first, and then we have the plasma membrane of the muscle fiber. And this endomysium provides electrical insulation, which means that when one muscle fiber is contracting, only that muscle fiber that receives the signal will contract and not the ones that are nearby. If you didn't have the endomysium, then once a muscle fiber starts contracting, then all the other ones will contract. So this endomysium is basically ensuring that only the muscle fibers that we actually need for that movement are going to contract. Let's say that I'm holding just this pen here. Of course, I don't need all the muscle fibers of a muscle to contract. For example, my bicep muscle here doesn't have to contract altogether because I'm only holding a pen. And so only a few muscle fibers of that muscle will contract to lift this pen up. And so this endomysium is preventing all the fibers to contract at the same time so that we can use only some of them, only the ones that we basically need for that movement. Okay, so if we take it in order, actually, let me add a few more things on top of the muscle. So here we have the muscle with the epimysium that we just see. And then here you see other layers on top of that, which separate the skin from the muscle. The skin, remember, is made by epidermis and dermis. And then afterwards, we have a subcutaneous layer. And then what we have is a superficial fascia. The superficial fascia is separating the skin from the muscle. But underneath the superficial fascia, we also have a deep fascia. And the deep fascia separates the individual muscles. So it's a layer on top of the epimysium that separates or join together muscles depending on the region of the body. Now, if you click on this link, what you're getting into is, and I feel like I need to explain this because if you click there, you're going to see a video of a person showing you how to take the skin out of chicken thighs. Now that is important because when that person is actually removing the skin from the chicken thighs are the muscles, you can actually see these superficial and deep fascia. Once you remove the skin, you can see that there's a very thin layer that is white, and that is the fascia. Okay, so that video is just to show you how it actually looks like. So you're basically separating the skin from the chicken thigh, which is the muscle, and so you will see these layers while you're pulling the skin out. It's also important because it's a skill that you might need in your life. And by the way, if you take the skin off chicken, or in general, when you use chicken, do not wash it. I know some people do that, but it's actually spreading bacteria around the sink. And so you don't want to wash it. You don't need to wash it. Once you cook it, it's going to kill all the bacteria. So don't wash your chickens. Okay, so here is another picture to show you what it looks like. So when you take a steak is usually not just one muscle, but multiple muscles. And so what you're seeing here is multiple muscles. There's one muscle here, there's another one here, and another one there. I don't know which muscle that is. I'm not a butcher, so I'm not really familiar with all the different muscles in steaks, but it's something interesting to look up. But anyway, so see here this white layer. That is the superficial and deep fascia. There's a lot of adipose tissue, so that's why it looks white. And it separates muscles, but sometimes it can also go over other muscles and keep them together. So it's just basically a branching out. And then here there's also epimysium, which you don't really see because it's very thin, but it's attached to the fascia, okay? And then if you look inside of this muscle, you can see that there are other layers here, what we call marble. Some certain steaks are better in taste because they have all these marble inside the steak. So here you see all these layers of white strands, and those are actually separating the fascicles. Okay, so here we have one fascicle, for example, there's another one here, there is another one here. You can actually see when you look closely, next time you have a steak, look closely before cooking, you're gonna actually see all these fascicles. And the fascicle is surrounded by perimysium. Now here we cannot really see the muscle fibers because they are really small. They are microscopic, so we cannot see them with the unaided eye. So next time you see a steak, look for the muscle boundary with the deep and superficial fascia and also the fascicles. Now these muscle fibers, remember, they are multinucleated. And that's because they actually come from single cells that fuse together to form one big cell. And so this is usually happening during embryonic development. The original cells are called myoblasts. And when they fuse together, they form a long fiber made with multiple nuclei that is called a muscle fiber. But there are some cells that are actually not doing that. They come from myoblasts. They decided to not join the other cells, but stay on the outer surface of the muscle fiber. And these are called satellite cells. These cells are actually really important because they help regeneration of the muscle fiber. So whenever there is damage to a muscle fiber, these satellite cells are able to produce new myoblasts that are going to fuse together with the muscle fiber to repair the damage. When you work out, these satellite cells are actually really important because working out also means damaging the muscle fibers. And this is they're called micro trauma, micro lesions of the muscle fiber. It actually helps the muscle fiber increase in size, and that will make the muscles look bigger. So whenever you're working out towards increasing your muscle fiber size, muscle size in general, then know that these satellite cells are involved in that process. They're going to make new muscle cells that are going to fuse together with the muscle fiber, increasing the size of that muscle fiber. Okay, let's take the muscle fiber. So we went down in the level of organization to the cell level. So this is a muscle fiber. Being a cell means that it contains very similar organelles than what we find in a regular cell. And so what we have here is the plasma membrane. Remember, the plasma membrane is under the endomysium. So we have the plasma membrane that in these particular cells is called sarcolemma. Whenever you see sarco, it means that it's related to muscles. So sarcolemma is the plasma membrane of these muscle fibers. Inside we find cytoplasm that for this cell is called sarcoplasm, but it's just the cytoplasm of the cell. And then see that we find multiple nuclei. There's one here and another one down there, another one here, and so on. Now, within this sarcoplasm, what we find is structures that are similar to what we've seen in other cells. For example, the sarcoplasmic reticulum is a modified version of an endoplasmic reticulum. Now, for this particular cell, the sarcoplasmic reticulum does a different function, which is storage of calcium. So within the sarcoplasmic reticulum that you see in blue here, we have calcium. And calcium is really important because it helps with the contraction of the muscle. We also have transverse tubules. Transverse tubules are basically invagination depressions of the sarcolemma that extend into the cytoplasm. And they help carrying the impulses, the electrical impulses that are coming from the sarcolemma and that are reaching all these myofibrils. So it helps also with the contraction of the muscle. When we look at just one myofibril, you can see that it's surrounded by the sarcoplasmic reticulum, as well as it's contacted by these T-tubule. It's a cylindrical structure made by myofilaments, which are strands of proteins. These proteins of the myofilaments there are two kinds. There are thick and thin filaments. Now you can also see that the myofibrils are divided into units called sarcomeres. So one myofibril is made by multiple sarcomeres, one next to the other ones. These are the functional unit of muscles. So by the shortening, the contraction of these sarcomeres will lead to the contraction of the entire myofibril. When you contract myofibrils, then the entire muscle fiber will contract. When you contract the muscle fiber, then the entire muscle will contract. But it all starts in these sarcomeres, which are subcellular structures. So let's take a look at these sarcomeres and what they are made of. So here we have the myofilaments. Remember, there are thin and thick myofilaments. The thick filaments are made by a protein called myosin. And you can see here, these are filaments of myosin. They have little heads that are protruding out of the filaments. While the thin filaments are made by a protein that is globular, not filamentous like this one, that it's called actin. For the purpose of this course, you just need to know that the thick filaments are made by myosin and thin filaments are made by actin. In terms of the contraction, you can see, first of all, that there is the sarcomere. This is what we're looking at here. The sarcomere has a central line called the M line to which the thick filaments are anchored to, while the thin filaments are anchored to another line here called the Z disc. We're going to see that next. But you can see that the thin filaments and the thick filaments are overlapping each other. Now, what happens is that these filaments will slide past each other towards the center of the sarcomere. And so they're basically going to shorter the length of the sarcomere. So the thick filaments stay in place while the thin filaments are going to come closer to this M line. You can see these animation here that is showing you just that. So here we have a sarcomere. We have the myosin with the myosin head. So this is the thick filament. And then these balls here are the thin filaments. So you can see that during a contraction, the thin filaments are coming closer together and almost touching each other at the center of the sarcomere. This is called the sliding filaments theory. And it was a theory because when they first hypothesized this theory, they actually couldn't see all these filaments. So they just looked at the sarcomeres, they looked at how they look like under a microscope, and they say, well, what if these lines that we're seeing are actually coming closer together? And that's how the theory happened. And then later on, this has been demonstrated from a molecular level by looking at these filaments and how they behave with each other. But basically what they've seen at first, and when they theorized that sliding filaments theory, is what you see here in this picture on the right. This is a histological picture of muscle fiber. What you're seeing here is the structure of the myofibrils. And you can see that it has different lines and different colors, and that's all due to the staining technique and also to the density of the proteins within those regions. So we can divide the myofibrils into segments called sarcomeres. And the sarcomeres go from one of these thick black lines to another one. So when they look at it under a microscope, they see a lot of these patterns all over these myofibrils. So one sarcomere is from this thick line to the other one. In the middle, there is this region called the M line that is surrounded by a zone called the H zone. Then you see that there is another band here, which is darker. Altogether, this is called the A band. And then this lighter band here is called the I band. And all these letters stands for words. That means that this region is lighter, this region is darker, and so on. We're not going to go into those details. They are weird words. So you need to know these organizations. So this is the sarcomere. It goes from one Z disc to another Z disc. In the middle, we have an M line. M stands for middle. Then around the M line, we have an H zone. Around the H zone, we have an A band. And then around the Z disc, we have an I band. One way to remember these is this mnemonic that I came up with. Mike has an invisible zoo or an invisible zebra, whatever you want. So you start from the middle of the sarcomere, Mike, so M line, then has, H band, then N, so A band. And by the way, zone and band are the same. Okay, so Mike has N, so A band. And then you go with invisible, so I band. And then in the middle of the I band, you put a Z disc for zoo or zebra. Okay, but remember that mnemonic, you kind of remember the organization of the sarcomere, and it will help you figure out all these bands that you see here. Now, next level, so once you master these and you know where all these bands and lines are, then you're gonna fill them up with filaments. And so basically we're looking at those bands and how they correspond to on a molecular level. Okay, so M line is basically the center of the sarcomere. This is where the thick filaments are anchored. And so the thick filaments you can see here, they form the A band. So if you go from beginning to end of a thick filament, then you have the A band. Well, if you go, if you look at the outside of the sarcomere, we have the Z discs. And then this region here is called the I band. Now the thin filaments are actually going all the way until the H zone. So if we're looking at the zone and the bands, we have the H zone, which is only made by thick filaments. We have the A band, which is made by both thick filaments and thin filaments. And then the I band, which is made only by thin filaments. Okay. So when there is a contraction, the sarcomere becomes shorter because the Z discs are getting closer to each other because the filaments are sliding past each other. The thin filaments are going to get as close to the M line as they can. So the H zone is going to become smaller. And so the overlapping between the thin and the thick filaments, it's going to get bigger, and that's the H band. Okay. So at this link, you can actually see what I'm explaining through an animation. And so you're going to see these filaments sliding past each other and how the A band, I band, A band, and H zone are changing through the contraction of the sarcomere. But just know that during a contraction, the H band and the I band are the only ones that become smaller, okay? And that's because these thin filaments are gonna go towards the M line, so the H zone that is only made by thick filaments are gonna become smaller. And then the same with the I band, these filaments by getting closer to the M line and the Z disc getting closer, then the I band will become smaller. Click on the link and you're gonna see it with your eyes. So this is a representation of a sarcomere when it's relaxed, and then at the bottom when it's contracted. And so you can see that the thin filaments are getting close to the M line, and that leads to the Z disc getting closer together, and that leads to the contraction of the sarcomere. And then when it's fully contracted, you can see that there is a big A band. In this case, the H zone is not there anymore, disappeared, because there is no area that has only thick filaments. And then at the same time, the I band has become very, very small because that's the region where there is only thin filaments. Also notice that the length of the filaments does not change. So the filaments are staying long. The only thing that changes is the relative position between these thick and thin filaments. So I like to represent these using my hands. So a sarcomere is basically from a Z disc to a Z disc, which are my thumbs. So my thumbs represent the Z disc, and then my fingers represent the filaments in general, not one or the other. And so when there is a contraction, the filaments are sliding past each other. And so you can see that the Z discs are getting closer together, right? But the length of the filaments is not changing. The length of my fingers is not changing. So they're just sliding past each other and making these Z discs closer together so that the sarcomere is also closer, is also shorter. And so it means that it's contracted. Now imagine that one sarcomere is here in one myofibril, but there's another one next to it and another one next to it. So you have multiple sarcomeres all contracting at the same time. So the entire muscle, the entire myofibril, and so muscle fiber becomes shorter. And so the entire muscle becomes shorter. Okay, so this is how the muscles are contracting and achieving a shorter length. So that's all because of the sarcomeres present in the myofibrils. Okay, so how does that happen? What is eliciting, what is triggering this contraction? The connection with the nervous system. So remember, in the nervous system, we talked about the somatic motor division, which is a division that is important for the motor control of skeletal muscle. The neurons that are achieving this function are called motor neurons. They are in the anterior horn of the spinal cord. So they are located in the spinal cord. And then they send their axons out through the anterior root of the spinal cord that merges together with the posterior into the spinal nerve. And then the spinal nerves are branching out in different directions. So from there, the axon of these motor neurons gets into the skeletal muscles of different areas of the body, depending on where they are in the spinal cord. So these are somatic motor neurons. They send their axon and axon terminal to touch or be close together to the muscle fibers. This type of synapse between a neuron and a skeletal muscle is called the neuromuscular junction. So this is the place of communication between the nervous system and the muscular system. In the neuromuscular junction, we have a synaptic knob, which is the expanded tip of the axon of this motor neuron. Within the synaptic knob, we have synaptic vesicles. That's similar to what we've seen before. This synaptic vesicle contains a neurotransmitter, which is just one kind for these motor neurons that are controlling skeletal muscles. This neurotransmitter is called acetylcholine, ACH. So the neurotransmitter used by motor neurons, somatic motor neurons that innervate skeletal muscle is always just one type, and it's called acetylcholine. Now, in order for the muscle to respond to that neurotransmitter, it needs to have a receptor. This is also true for other cells, for example, the one that respond to hormones. In order to figure out that there is a hormone, that there is a molecule, they need to have a receptor. So the skeletal muscles have receptors for acetylcholine. And these receptors are in this region of the muscle called the motor end plate. Motor end because this is the end of the motor neuron, and plate because it looks like a plate, like a little bowl. There is a space between the motor end plate and the axon terminal called the synaptic cleft. The synaptic cleft is where the acetylcholine is released through vesicles, and it's where it binds to the acetylcholine receptor on the skeletal muscle. And then you see here briefly what it does. Once the acetylcholine receptor binds to the, once the acetylcholine binds to the acetylcholine receptor, then it leads to a change in the electrical activity of the muscle fiber, which is transported by T tubules throughout the muscle fiber, and that leads to the release of calcium from the sarcoplasmic reticulum. That calcium is going to go to the myofibril, and it's going to be really important for the contraction of those myofibril. And so you can see another version of what I just explained here. There is an electrical impulse coming down the axon of the motor neuron. This is usually decided by the central nervous system. So let's say that, okay, I need to scratch my face. So there is an impulse coming from my brain down the spinal cord to those motor neurons that are controlling my hand. And then they send an electrical impulse to the neuromuscular junction of the muscles that are important for moving my hand to my face. And that leads to the release of acetylcholine to those muscle fibers. Now there is the acetylcholine binding to the acetylcholine receptor. That leads to an electrical stimulation of the muscle fiber that leads to the calcium being depleted from the sarcoplasmic reticulum. So the calcium is expelled out of the sarcoplasmic reticulum. And here, we're not going to go into these molecular details, but the calcium is important so that the filaments are basically sliding past each other and leading to the sarcomere becoming shorter and the entire muscle becoming shorter. Now, in order to have relaxation, which is what you see on the right side, you need to remove the calcium. So there are going to be some pumps here that take the calcium from the sarcoplasm back into the sarcoplasmic reticulum, and that leads to the relaxation of the muscle. And both contraction and relaxation, they need ATP. And this is why when someone dies, the muscles of the corpse, of the cadaver, they remain stiff, so in a contracted state, because they require energy to relax, to get the calcium back into the sarcoplasmic reticulum. And so they stay stiff because they don't have the cells don't have energy because the person has died. And so they cannot use oxygen, they cannot use glucose. These muscles relax only when the cells start to degenerate. And so the filaments are starting to degenerate, to degrade, and so everything basically is falling apart, all the structural organization of the muscles, and so at that point they relax. Okay, so motor units. Motor units are what we collect together as one motor neuron, so a single motor neuron, and all the fibers that it controls. So one motor neuron can control multiple fibers. Within one fiber, though, there is only one connection with the motor neuron, so only one neuromuscular junction per muscle fiber. But one motor neuron can innervate multiple muscle fibers, okay? So a motor unit is one single motor neuron and all the muscle fibers that it innervate. Now the motor unit size, so having a lot of muscle fibers for one motor neuron or having very few muscle fiber, depends on the muscle. So there are muscles that have large motor units and muscles that have very small motor units. And it all depends on the action of that muscle. So let's say muscles of our thighs, those are probably going to have very large motor units because with just one electrical impulse, we want a lot of muscle fibers to contract. But muscles of our eyes, for example, that have very fine movements, or fingertips, then they might have small motor units because we want just few muscle fibers to contract when we send that signal. There is a principle called all or none, in which when one motor unit is stimulated, so when there is an electrical signal coming from a motor neuron, then all the muscle fibers within that motor unit will contract. It's called all or none, meaning that either all muscle fibers are contracting or none of them. If none of them, it means that the motor neuron is not sending the signal. But if the motor neuron is sending the signal to the muscle fibers in its motor unit, then all of them will contract. Now, the muscles are attached to bones through strands of connective tissue called tendons. So tendons are attaching muscles to bones. We're going to see that there are also strands of connective tissue attaching bone to bone, and those are called ligaments. So for muscles, we have tendons that attach them to bones, and this attachment is the cause of the movement. So when a muscle is contracting, then the bones are coming closer together, and that leads to the movement. So for example, here we have this muscle. It's attached to the shoulder joint, as well as the radius of the forearm. And so when this muscle is contracting, then the radius is going to come closer to this other bone called the humerus. And so they're going to come closer together, so the arm is going to flex. Okay, when the muscle is relaxing, you have the opposite movement. Now, in terms of muscles and how they are attached to the bones, we have usually one that it's the less movable attachment called the origin, while the insertion is the more movable attachment. And so, for example, in this muscle here, this region is not moving when the muscle is contracting. So this is the origin of the muscle. This is where the muscle is attached to the bones. And then we have these other attachment, which is more movable when the muscle is contracting. This radius is moving. And so that is the insertion of the muscle. These days, these are also called superior or inferior attachment, or proximal and distal attachment. The muscle tone is the constant tension that is present even in a resting muscle. So motor units are usually stimulated even if you're not moving that particular muscle, if you're not contracting, to avoid fatigue. But there's always a muscle tone. There's always some muscle fibers that are contracted in that muscle. Now, the contraction can be isometric or isotonic. Isometric means that when there is the contraction of the muscle, the length of the muscle remains constant while the tension is changing. So for example, in this case, isometric contraction, this person is lifting a weight, but it's not moving the arm. So the length of the muscle remains constant throughout the contraction, but the tension of the muscle changes. This is usually done in type of exercises in which you are basically holding a pose, for example, a static pose. So for example, you're lifting, but you're not doing any other type of contraction, flexion or extension. So you're just lifting something. You're holding your grocery bags, right, with your arms straight down. You're still contracting the muscles to hold that weight, but you're not actually doing movement with those arms. Holding a yoga pose, it's usually isometric type of contraction. If you're trying to push on a wall, that's also isometric. A lot of climbing, carrying objects is an isometric type of contraction. While isotonic, it means that the tension is constant and the length of the muscle is changing. So, for example, when you're lifting weights and doing concentric contraction, which means that you're pulling the weight closer to you, then the length of the muscle is changing. And so this is called isotonic contraction. And usually when we're walking, running, when we're doing weightlifting, push-up, biceps curls, those are all exercises that require a type of isotonic contraction. Concentric contraction within the isotonic means that the muscle is shortening, while eccentric means that the muscle is lengthening. Now, in terms of actions of skeletal muscles, We have agonist and antagonist. Agonist is when the contraction is produces with the movement. So this is called the prime mover. So for example, the triceps brachii, the agonist of forearm extension. When you're extending, you're using the triceps brachii to do forearm extension. That is the agonist. The antagonist is usually on the opposite side, and it's a muscle that the action is basically opposing the one of the agonist. An example is the biceps brachii. If an agonist produces extension, then the antagonist produces a flexion. There's also synergist muscles that are muscles that help with the they help agonists performing an action. So for example, here with forearm flexion, we have the brachioradialis and the brachialis that are synergists of the movement. And the biceps brachii is the prime mover. So they are just helper. I'm gonna stop here with muscle naming. We're gonna move on to other muscles. Thank you for watching.